All right. Hi, my name is Justin Rogers, and I'm going to tell you about the awesome research that I did with Dr. Renberg and Dr. Pennington at the Army SPARK program. Okay, so starting up here, factors influencing clearance phenotype of fungal isolate from the environment. I promise this is more interesting than that sounds. I'll go through this pretty quick. So plastic, the good part about plastic is that it lasts for a long time. So for this project, I focused on polyurethane ester. So that's just one type. Polyurethane, if you left it outside, it's going to be there for like 100 years. It takes forever to decompose. Now, what's interesting about that is that there's actually fungal organisms that in starvation conditions will eat plastic. Now, it has to be starvation conditions because, again, if there's anything better in the area, they're going to eat that, which makes sense to me because I wouldn't want to eat plastic either. My organism was named 1058, and basically what I was tasked with was, can I augment this organism's ability to interact with, probably eat, plastic? So we used waterborne polyurethane, which is called Imprenil. We made these plates. By the way, I made hundreds of these. And it makes this white color on the plate. So if you look down here, you can see this white color. This is the area that has been interacted with, probably ate by the organism. This is the area that hasn't. So what's fascinating about this, just like I mentioned, we're talking 100 years for polyurethane. This organism is doing it in like 4 weeks. So that's amazing. That's way better. I looked at 3 different parameters. I changed all the media types. And what you can see is that the media definitely influences the type of morphology that this organism makes. You can see at low nutrient, there's basically nothing. And high nutrient, it makes these really organized structures. We also see a lobate morphology, which is like lobes. And we also see this phenotype that I affectionately refer to as the communion wafer. It's a series of circles and crosses in the middle of it. We don't really know much about this organism, and we actually have no idea why it's doing that. So definitely room for further research. The other thing I looked at was temperature and supplementation. So when it came to temperature, the organism did not like 12 degrees Celsius, which is pretty cold. The organism loved 22C. That makes sense. I'm a California boy. I love 22C. That's about 71 degrees. And 32 degrees was a little bit hot. So the organism still was able to thrive and survive, but not as good as it did at 22C. The other thing that I did was I added some nutrients to it. So I added L-serine to the plates to see if that could augment this organism's ability to interact with plastic. And what I found was the opposite. So I was really wrong. If you add L-serine, it's going to reduce this organism's ability by 80%, which is pretty astounding. That means that the organism is eating the L-serine as opposed to the plastic. Because again, starvation conditions. If there's better stuff, it's eating the better stuff. It doesn't want to eat plastic. Next, I added ammonium sulfate. And ammonium sulfate increased it by 20%. So what this means is that the organism was most likely using it as a nitrogen source to produce these enzymes and also to build bigger colonies. So what are the potential impacts of this? Well, the Army could produce materials with fungal spores in them. What this means is that we would have plastics that have all the benefits of plastic. It would last forever, be shelf stable, and temperature resistant, all the things that we love plastic for. But when we want it to, and that's the key, when we want to, we could cause the plastic to rapidly degrade, again, in the course of days, if not weeks. What this could mean is single-use technology for soldiers. So soldiers could have tools with this built into it. When they get to the job or whatever they're doing, they use the tool and then they leave it there, it decomposes. The thing that I thought about was 3D printed drones. They would be able to fly the drone to whatever location they wanted, crash it, and then trigger something and it would decompose. But other than that, it would behave like normal plastic. Thank you so much for listening.